This is Mr. Wartsy again, and this is the fourth screencast of plants. Last time we were here, this is the last slide that you looked at. This was the stem longitudinal section of a stem. Why has I said that? I repeated myself. Sorry. So a leaf is the site of photosynthesis. The petiole is the stem, and the blade is the flat portion of the leaf. The dicot usually has a large midrib and branched veins from that midrib, while monocots have parallel leaf veins. Now, the upper epidermis, the lower epidermis, guard cells, stoma, palisade, mesophyll, spongy mesophyll, bundle sheath, xylem, and phloem, those are all the things that we're going to talk about when we take a look at the dicot and monocot leaf cross section. So we have the upper epidermis here and lower epidermis, and they pretty much just protect the leaf. Whereas you have the palisade layer here, these cells are parenchyma cells, and they are filled with chlorophyll molecules, so they photosynthesize. Now the spongy layer, the spongy mesophyll here, not much chlorophyll, and you'll notice they have all these air spaces, and that allows for gas exchange. So oxygen is going to leave the leaf and carbon dioxide is going to enter. You're going to ask yourself how. In the bottom here, you're going to see stomas. We don't really see stomas here. And the stomas are controlled by guard cells. You see stomas here, you see stomas here. This is a monocot, different situation we'll talk about in a second, but they see them in dicots too. And here is the vein. This is a major leaf vein, and we see xylem, which are the big cells, and the phloem, which are the small cells. Okay. Now these things are surrounded and protected by the bundle sheath cells. Right. Now here is a monocot. You notice the monocot, there's not really much of a palisade layer. This is pretty much all spongy mesophyll. The very thin, not very thick, and you can tell it's a monocot because all these leaf veins are parallel to each other. There's a row of leaf veins. Big cells are xylem, small cells are phloem, and we have the bundle sheath surrounding it. So not much of a palisade tissue. Instead, the spongy layer does two things, gas exchange as well as photosynthesize. But we see guard cells, and let's see if I can show you right here that we got the stoma. Take a look at the bottom stoma. There we go. And we got the guard cells on either side here, which allows for gas to be exchanged. And we're going to take a look at root stems and leaves in your slide activity. So we finished the parts portion, the anatomical portions. Now we're going to talk about how these plants actually survive. And we need to talk about the transport, how water moves, how the photosynthetic product moves, and how gas is transported. And we're going to spend some time on water. And when we talk about water, we need to talk about short distance and long distance transport. So the short distance is how water gets from the environment to the xylem, and long distance is how water gets from the roots all the way to the leaf where they're needed for photosynthesis and where transpiration occurs. So short distance, we need to talk about three different ways that water can move from the outside into the xylem. The first is osmosis, the movement of water from a high water potential to a low water potential to a semi-permeable membrane. We've talked about that before. Then water can move from the high water potential to the low water potential all the way through to the endoderm. It's going to stop there. The symplastic movement is the movement of water through the plasmodesmata. Remember those channels that connect each plant cell? And the water moves from plant cell to plant cell to plant cell through these channels. And apoplastic is that the water will move through the cellulose, the fibrous cellular cell wall by adhesion and cohesion and keep on moving until it hits the endodermis. So water enters the root hairs right here. Then the endomycorrhizae can play a huge role in absorbing the water, and those are the zygomyces that increase the surface area to volume ratio. Then it'll move by osmosis, symplastic or apoplastic movement to the endodermis. Now that's important because there's a Casparian strip that surrounds the endodermis. Remember that? It's that waxy layer, that suberin. Now, since water can't go through the suberin, the water is actually going to have to enter the plant cell and move through the endodermis by the symplastic movement. It has to move through the plasmodesmata to get through the endodermis. Now, this allows the plant cell to select what enters the xylem. So right here we get the epidermis, the cortex, right? The endodermis. Now see how it's moving through. This is symplastic. Now if it's moving through the cell wall, that would be apoplastic. And it moved through here, that would be by osmosis. Now right here, the suberans here. So everything's going to have to move through the endodermis by symplastic movement into the pericycle, which will then shove the water into the xylem. Long distance. So the water entering pushes the water up in front of it, up the xylem, right? So that can only work for so long because there's going to come a time when the gravity is pulling down on the water and the water can't be pushed up anymore. Now the water forms chains in the xylem through adhesion and cohesion, specifically cohesion. It'll try to climb up the xylem by adhesion, but it won't be able to fight gravity for too long. But that's called root pressure, the height of how high the water can travel up the tree or the plant just by simple pushing of the water molecule behind it. Now, if the water gets to the leaves, right, the water will evaporate in the leaves. That's called transpiration. As the water leaves the leaf, it will pull by cohesion on the water molecule behind it. And as it pulls on the water molecule, the other water molecules will move up the xylem. And that's called the transpiration pull. So here we have the being pulled up the xylem tubes. The water evaporates over here, and as the water evaporates, it will pull the water up the xylem. Now, every leaf has all the xylem going to it. So every leaf has xylem and phloem coming and going from it. So every time you see a big leafy tree, all those leaves have all the xylem, and all that xylem is in the stem, and all that xylem goes to the roots. So what can affect transpiration? And there's an AP lab on this, and we'll look at it, but temperature, air currents, and humidity. Basically, anything that affects evaporation will affect transpiration. So temperature and wind will increase evaporation, so therefore it will increase transpiration. Humidity, on the other hand, high humidity will lower evaporation. It will lower transpiration. Now, guard cells on the bottom layer of the leaf will also affect transpiration. How? Well, when the guard cells are open, allowing gas to be exchanged, then the water can evaporate. If the guard cells are closed, the water cannot evaporate, so it will stop transpiration. Which brings us to the next type of transport, which is gas exchange. So guard cells have an unequal primary cell wall. They are thicker on the inside. So when guard cells fill with water, they will swell around that thickened cell wall, and they will form a hole in the middle of it, which is called the stoma. And I can show you in class if you really want to see that tomorrow. Now, potassium will affect water movement. If you increase potassium into the cell, the more water moves in by osmosis. You decrease potassium and the water moves out by osmosis. So what holds the potassium but the large central vacuole? The large central vacuole, that big vacuole in the plant cells, it contains potassium besides other things. And the tonoplast, which is the membrane around the large central vacuole, will control the movement of the potassium into the guard cell or back into the large central vacuole. If the potassium moves from large central vacuole into the guard cell, into the cytoplasm, water moves in. When the potassium moves out of the cytoplasm back into the large central vacuole, water moves out. So right here, this right here, the inside here will have, do you see how this is really thick here? That's the thick cell wall. So the whole cell will expand around that thickened cell wall, and as it expands around the thick cell wall, it forms a hole in the center called stoma. Okay, so water moves in because of the potassium ions entering the guard cell from the large central vacuum. So there are plants that have adaptations to really dry environments, and those are called xerophytes. They live in arid environments. They usually have small, thick leaves, thick cuticles. The cuticle is the wax cutin on top. Stomas are found in crypts, and that will decrease water loss. So crypts are little pits, and the stomas are found in those pits. They open the stoma at night, right? So it's cooler at night, so it decreases water loss. They store carbon dioxide as malic acid during the day, so they can produce glucose by using the stored carbon dioxide at night. And this is called Kraslucian acid metabolism, the production of the glucose at night. Food transport, which is also called translocation, is known as the pressure flow hypothesis, which is a source to sink. So you move it from where the glucose is produced to where the glucose is needed. So the sugar will enter the phloem by active transport, right, and it produces a hypertonic environment, low water potential. Water from the xylem will enter the phloem by osmosis by moving from a high water potential to a low water potential. As the water moves into the phloem, this increases the water pressure in the phloem and forces the sugar down the phloem. Now, when the sugar is needed, sugar will leave by active transport. There's a decrease in sugar content, which means that there is an increase in water potential, so the water will leave by osmosis back into the xylem. You need to remember, the xylem still has minerals and other salts, so it's not pure water that's found in the xylem. So we have here, the phloem is on the right, so we produce the glucose. Glucose is moved into the phloem by active transport. This decreases the water potential here. Water flows from the xylem into the phloem, increases hydrostatic pressure, which forces the fluid down. As it forces the fluid down, whatever cell needs the glucose will take it by active transport so they can produce energy. So this is the source, this is the sink. Moves from source to sink. Plant behaviors. Darwin and his son Francis Galton, we can talk about him later on, but he wasn't a very nice guy. Plants grow towards the light. If you cut off the tip of the plant stops growing towards light. So they're thinking there must be something in the tip that causes it to grow towards the light. Peter Boysen Jensen, he cut the tip off and placed gelatin between the tip and the rest of the stem. And guess what? That tip still grew to the light. The plant grew to the light. But if you placed mica, which is a very thin rock, between the tip and the stem, it did not grow towards the light. So there was something that was produced in the tip that moved through the gelatin into the stem that caused it to grow towards the light. And F.W. Went proved this because he took Peter Boysen Jensen's experiment one extra step by cutting the tip off and putting this gelatin on the tip, and it grew towards the light. We have it here. Goes towards the light. Doesn't grow towards the light. Now we cover the tip by opaque cap, doesn't grow towards. Transparent cap, it does. Okay. So here's the gelatin here, and here's the mica there. Yeah. Now FW1 took this little piece of gelatin here, put it on top of this one, and it grew towards the light. So it's something that was produced by the tip that moved through the gelatin to cause it to move to the light. So what causes it to happen but hormones? Plants have hormones, and we are going to talk about the plant hormones next time we get together. Well, I hope this helped, and have a good night.